Osmosis. What is osmosis? It is the movement of water molecules from their region of higher concentration. Higher concentration means that has more molecules of water as compared to the solute, to their region of lower concentration. That means where there is less concentration or less molecules of water as compared to the solute, and very important, through semi-permeable membrane. Okay, they are not in direct contact, and it is only the movement of water molecules, no other type of molecules. Now, important thing to note is that only water molecules will move from their higher concentration to their lower concentration through semi-permeable membrane. Now, if children write that osmosis is the movement of water molecules from dilute solution or dilute solution to concentrated, or if they write more water molecules from higher concentration to lower concentration, then you are not specifying whether you are talking about the solvent or the solute. So you have to be very clear that water molecules from their higher concentration to their lower concentration. Now there you are referring to the term water. Okay, so with reference to cell, now osmosis can be either way, inwards or outwards, depending upon the concentration of the solution which is surrounding it. So we have two types of osmosis. One is end osmosis, another is exosmosis. End osmosis, that means osmosis is taking place but it is inwards. That means water from outside is moving inward towards the cell through semi-permeable membrane, and this will tend to swell up the cell. So end osmosis, inward movement of water molecules. Exosmosis, outward, exit. You can also understand from this, that is the movement of water molecule through semi-permeable membrane towards outside, and this will cause shrinkage of the cells. So if the concentration of the surrounding solution is the same as that of the outside, so in which direction will the net movement of water molecules be? So inward, outward, no, there will be no net movement. Now this is an experiment which will help you to understand osmosis. Now see, in this experiment what is done? Now this is a thistle funnel, and this thistle funnel is filled with sugar solution. That means this solution has water molecule and sugar molecules, but more of sugar molecules. Then this is a cellophane paper which is going to act as a semi-permeable membrane. So we have covered this part with cellophane paper, and this beaker is filled with water. Hardly any sugar molecules are there in this beaker. Now if you leave it for some time, after few hours, what are you going to see? Now this was the initial rise, the level will rise. Why there is rise? Because the water from the beaker will move through this cellophane paper, which acts as a semi-permeable membrane, towards the thistle funnel. And so as the water enters here, increase in the water molecules will lead to the increase in the level. Okay, but you will not see any sugar molecules coming out, because sugar molecules will not pass through the cellophane paper. So take some concentrated sugar solution in a thistle funnel, cover the mouth with a cellophane paper. You also have another example, egg membrane or animal bladder, which also act as a semi-permeable membrane. Invert the thistle funnel which contains in a beaker which contains water. After some time, this is now experimental setup. After some time, you see the rise in the level of the. Now as a control for comparison, you will take another funnel with plain water filled in it. Now instead of sugar, we will have plain water. Again, mark the level on the stem, so you will not see any rise in the level in the stem. So the sugar solution in the thistle funnel will rise, and the level of water in the control setup will remain unchanged. So you also should know what is a control setup in which the experimental point is missing. What you are studying is missing. So the level in the beaker in the experiment will drop slightly, while the one in the beaker in the control will remain unchanged. Now, level in the water. This is also very, very important point. See, if you get such a question and they are telling you that redraw it, if this is your initial level and you are showing a rise in the level after few hours, then this level in the redraw should come till here. Okay, that mark, this is the level of the water where it will reach. If you will show the same level, then the answer you will lose marks. If the water in the beaker in the experimental setup is tasted, it is not found sweet, again because sugar molecules cannot pass through this cellophane paper. Now, some water of the beaker has passed through the cellophane paper to enter the thistle funnel containing the sugar solution. Sugar from the thistle funnel has not passed, and the cellophane paper act as selectively, we don't say semi-permeable, selectively or differentially permeable membrane. It allows only water molecules to pass and not the sugar molecules. So this is the main three conclusions, and you need to learn these three conclusions. Then modified version. Now in this, instead of us by taking more concentrated sugar solution in the thistle funnel and less concentrated sugar solution in the beaker. See, highly concentrated sugar and less concentrated sugar. Again, the answer is going to be same, that there will be rise in the level of this capillary. Why? Because more water molecules outside and less molecules inside. So it's going to enter into the thistle funnel. The level of the solution in the thistle funnel would rise. This is again because some water from the less concentrated sugar solution move inside the more concentrated sugar solution through cellophane paper. Now instead of cellophane paper, if we will take a rubber sheet or we take muslin cloth, what is going to happen? So just use your logic here. The rubber sheet is impermeable, would not allow any water molecules to cross over. That means there is no rise in the level. And muslin cloth, there are so many books, they are large pores, so many pores and large insides, so they will not hold this sugar solution. You will see the mixing of the water and this sugar solution, and the entire sugar solution will flow down to a common level in the gravity. Therefore freely permeable, muslin cloth is freely permeable to the sugar solution. The same experiment can be performed by taking the whisking bag. It also acts as a semi-permeable membrane. Now you can see here, whisking bag. So water from all the sides of this bag will enter, and there's going to be rise in the capillary tube. So because whisking bag is also acting as a semi-permeable membrane. And then there are two key points, that if there are two different regions, we call them as tonicity. Okay, so 2 points you mark, the 2 regions of different concentrations of water molecules or 2 regions of different concentration of sugar molecules. And a semi-permeable is one which allows only the passage of selective molecules to pass, that is the water molecules, and prevent the passage of the solute.